Funding for this program is provided by Annenberg CPB to advance excellent teaching.
Advances in molecular biology are facilitating unprecedented changes in living organisms.
There's a huge potential of what you can use biotechnology for, and it's not just plants.
Genetically modified organisms are revolutionizing agriculture, medicine, and industry.
Hey, EPA, don't do what I meant to say!
But critics charge that technology is moving too rapidly with too little regulation.
We really don't know what the long-term effects of most GM products are.
What are transgenic organisms? How are they made? And how are we using the organisms we create? Humans have been manipulating plants and animals for nearly 10,000 years. Throughout that time, people have selected plants and animals that have most closely fit their need, then planted or bred them to improve their stock. Over time, this practice produced change in the genetic makeup of the organisms. These corn cobs found during excavations in Mexico are examples of evolutionary change. Primitive corn dating from about 5000 BC is much smaller than the more recently cultivated ears dating to about 1500 AD. At biotech companies and universities like this, scientists are using the tools of biotechnology to move genes within species to change their traits and characteristics. The corn we enjoy today, for example, is a product of decades of artificial pollination or conventional breeding to produce better yielding hybrid plants. But now there's a new way of rearranging genes quickly, precisely, and from one species to another. It's called genetic engineering. Dr. David Dornbos of Syngenta Seeds was involved in the testing of genetically engineered corn in the early 1990s.
Conventional breeding is very much a broad-based approach. You're literally recombining the whole corn genome at the same time. When you make a cross, you're recombining hundreds of thousands of genes. And you may be selecting against 15, 20, 25 traits at the same time. So in the case of genetic engineering, again, we're recombining DNA, but now in a very targeted sense. You're taking a very specific gene from, it could be now a soil bacterium like Bacillus thuringiensis, and you're combining it into the corn genome, but now you're crossing the lines of two different species.
Bacillus thuringiensis, better known as BT, was first isolated almost 100 years ago. The bacterium produces crystal-like proteins that kill specific insects that eat them.
There are thousands of different types of BT proteins, and as they form crystals in the guts of insects, for example, each different protein forms a slightly different type of crystalline structure. And those different BTs then become very selective for different types of insects.
Powdered BT was registered with the USDA as a natural insecticide in 1961. It's used primarily by organic vegetable farmers, but because it's biodegradable, it creates economic problems for large-scale corn production.
You're literally putting the protein from BT on the surface of the plant. It's exposed to the rays of sunlight and is broken down fairly quickly. So the shelf life of effectiveness on the plant then is relatively short.
Because of its limitations as an applied insecticide, scientists began to think about using BT in a different way.
They thought, hey, maybe this is kind of a slam dunk, ready-made case of applying a biotechnology combination of tools to get this thing to work in a corn plant. Just working with DNA extracts from Bacillus thuringiensis, they were able to isolate out the single gene that caused the end results.
The tools of biotechnology start with the construction of a recombinant DNA molecule, the insertion of DNA from one species into a plasmid vector. The first step is to identify a gene of interest, in this case, a pest-resistant gene from the BT bacterium. In creating recombinant DNA for plants, the most commonly used vector is a plasmid isolated from Agrobacterium tumefaciens. Next, restriction enzymes, which recognize specific DNA sequences, cut both the plasmid and the DNA of interest between corresponding base pairs.
So the restriction enzymes are a way of getting down to smaller pieces, more manageable pieces of DNA and even single genes.
Any two fragments of DNA cut by the same restriction enzyme can then be recombined because they have complementary sequences on their dangling or sticky ends that pair back up.
The other part that you need is a series of promoters and controllers so that, you know, when the gene is in the genome of the corn plant, it's turned on. The third part that they needed was, again, just another type of a marker, and we used as a second marker a herbicide-resistant gene.
During transformation, the recombinant plasmid with the gene of interest can be reintroduced into Agrobacterium and cloned as the bacterium multiplies naturally. The bacteria can be used as vectors to infect plant cells growing in culture. Or the DNA construct can be introduced directly into plant cells by a variety of methods, including the gene gun method. The transformed or infected plant cells can regenerate a new transgenic plant in vitro because the cells are totipotent. Each cell has the ability to form all parts of an organism.
The fact that corn cells or plant cells are totipotent, which means that they have the potential to express all genes, does make it easy to produce a callus tissue from a few cells, and with plants that's certainly an advantage. And so by focusing on trying to build effective Bt corn, that actually drove the development of many of the tools of science to get you there.
Today, corn is the single largest crop in the United States, and almost a third of it is genetically engineered. Len Corzine's family has been growing field corn in central Illinois for 5 generations. Like most farmers, he buys new seed every year and plants a variety of hybrids to try and optimize his yield. In the late 1990s, industry began offering farmers the option of Bt corn.
We looked at the Bt corn as a new tool in the toolbox, and with the seed being more expensive, we didn't try a lot of it first, but we tried a little bit just to see what it would do.
Bt corn protects against an insect called the European corn borer, or ECB. The larvae cause more than $1 billion of damage to the U.S. corn crop every year.
This is a typical corn borer damage where the corn borer has gone into the shank and basically killed this ear, and it has dried up prematurely. You can see it next to one that has not been affected, but the corn borer caused this to die.
At Syngenta Seeds, scientists brew a special artificial diet to raise corn borers for research. The larvae test whether a new transgenic plant is expressing the Bt gene.
If the corn plant is expressing Bt, those larvae will die within 24 to 48 hours. So you have a very quick screen that way. This particular technology trait is very, very effective against the ECB. So the control rate is 99.9% plus.
Prior to Bt corn, farmers protected their crop with chemical insecticides, but the process was expensive, labor-intensive, and required precise timing to work. Many farmers say Bt corn is better.
It's just a better way, a more environmentally friendly way, because say if I had to spray around my house with my kids here, or I had a neighbor if I'm next to a development, I don't want to be spraying a toxic chemical next to my home or anybody else's home for that matter.
For most farmers, the final decision to use Bt corn is driven by economics, especially in areas of high corn borer pressure and periods of low crop prices.
You can be very precise, and at the end of the day, you provide a higher quality product using less chemicals, and it has to be more economic or it won't last. That's the bottom line.
While some plants, like corn and soybeans, are engineered to resist pests, other plants are being genetically transformed to improve their nutritional value. The Rockefeller Foundation in New York City has a long history of supporting research in rice biotechnology.
The polished rice, which is what people consume, totally lacks vitamin A. It also lacks the precursors of vitamin A, beta-carotene and other carotenes that can be converted to vitamin A when other foods are consumed.
Approximately 3 billion people worldwide depend on rice as their staple food. But that can result in vitamin A deficiency, which can cause health and vision problems, especially in children. According to the World Health Organization, up to 500,000 kids go blind each year.
But in addition to that, there's good evidence now that mucous membranes throughout other parts of the body have been weakened, which makes the body more susceptible to other diseases such as measles.
Like all green plants, rice plants produce beta-carotene in their green tissues. But no rice plants produce carotenes in the endosperm. But the grain does contain the compound geranyl diphosphate, or GGPP, which is a precursor to carotenoid production.
That meant that we would need to use genetic engineering approaches where you actually added genes to the plant in order to produce the beta-carotene, or somehow figure out how to turn on the genes that were already in the rice plant in the grain tissue where they were not normally turned on.
With funding from the Rockefeller Foundation, 2 European scientists, Professors Ingo Potricus and Peter Bayer, proposed turning on the GGPP through 4 enzymatic steps.
Nobody had introduced 4 genes into a plant and gotten them to function in a sequential way that allowed a whole biosynthetic pathway to be introduced previously. So it was cutting-edge research.
From prior research, scientists had already isolated 2 genes from the daffodil, encoding for 2 of the enzymes required for turning GGPP into beta-carotene. Each of the genes were modified by adding specific promoter and leader sequences, so the genes would only be expressed in the endosperm.
When Professor Potricus added the phytoene synthase gene from daffodil, and the rice plants produced phytoene, that was very encouraging because that was the first step in the pathway.
Next, scientists introduced the gene for the second enzyme, phytoene desaturase, isolated from the bacterium Erwinia. This gene carried out the 2 steps necessary to convert phytoene to lycopene. For the final step, the second daffodil gene produced the enzyme lycopene cyclase, which converted lycopene to beta-carotene. In combination, 2 daffodil genes and 1 bacterial gene produced the enzymes that carried out the 4 steps to convert the precursor to beta-carotene.
And it turned out that was the breakthrough that they needed. This technique of introducing all of the genes together at one time led to the first of the rice plants that produced grain that had the desired golden color. And then Peter Bayer was able to show that golden color was in fact beta-carotene.
At the International Rice Research Institute in the Philippines, golden rice is undergoing extensive safety and nutritional testing. One goal is to cross golden rice with well-growing native varieties. The new breeding lines will be released free or at minimal cost to poor farmers in developing countries.
This has been our vision from the beginning. The idea behind the golden rice is to distribute it only once and then allow the local farmers, subsistence farmers, to multiply, to keep their seeds for further sowing so that this problem does not occur anymore.
The inventors of golden rice, the Rockefeller Foundation, and various biotech companies are collaborating to help speed regulatory approval and distribution.
I think this is a good example of where the private companies recognized that this was not a product that was going to have great economic value, but that would have great social value, and were prepared to contribute to make that happen.
Golden rice currently contains very low levels of beta-carotene. This is about 10 ounces of rice, the average amount that most people in developing countries have to eat every day. Researchers hope to produce golden rice that contains enough beta-carotene to have a beneficial effect in one such serving. However, golden rice still faces many months of research before it will ever be available to farmers. While some say biotechnology offers great hope for the future, others say the technology is outpacing our knowledge of the implications it may hold.
Hey, EPA! Don't
Protesters in the U.S. and Europe are charging that biotech companies are putting profits before safety and demanding a global moratorium on GMOs, products they call frankenfoods. Dr. Rebecca Goldberg of Environmental Defense says there are too many unanswered questions and too little regulation.
We really don't know what the long-term effects of most GM products are. My guess is that many of them are going to prove perfectly safe, but there's plenty of reason to think that some of them may not be, and certainly if we begin to see more and more GM foods on the marketplace in greater variety, I'd bet that some of them are going to cause some problems.
Corn and soybeans are the most heavily genetically engineered food crops and are found in more than 70% of processed foods. That's of prime concern to people with allergies.
In particular, all known food allergens are proteins. So every time a new protein is added to a food by genetic engineering, there's a small but real chance that an allergen is being added to that food. But in the case of genetic engineering, because there's no labeling of genetically engineered foods, even if people do at some point begin to develop allergies to, say, genetically engineered soybeans, they're not going to be able to avoid them in their diet because they can't know what they're eating.
While food safety is one issue, GE crops are at the root of many environmental concerns as well, from the effects of toxic pollen to the possibility of superweeds.
In certain circumstances, crops may transfer a trait such as disease resistance that actually confers a new advantage to a weed, makes them more abundant, makes them more of a management problem. That threat is conjectural at this point, but certainly is biologically entirely reasonable. Another big issue with Bt corn and other Bt crops is that widespread planting of these crops, which have Bt toxins in their tissue all the time, whether or not insects are feeding on a plant or causing economic damage to a crop, is that the use of these crops will lead insects to evolve resistance to Bt toxins.
Concerned about the possibility of widespread resistance, the EPA is requiring farmers like Lynn to set aside up to 20% of their crop for non-Bt corn in an insect-resistant management plan, or IRM.
The fear is that if you only have Bt corn, even though you've got a high-dose strategy, you end up with an insect race of European corn borer that are fully resistant to Bt corn.
By allowing corn borers to safely breed in a refuge of conventional corn, the genes for overcoming resistance will be diluted, slowing the evolution of resistant insects. Farmers must plant the refuge within a half-mile of their Bt fields.
That's a good idea because I think it's showing that the industry and agriculture, if you will, is being responsible, and we need to know what those rules are, and it is our responsibility to play by those rules.
We do keep track of the counties in which we sell Bt and non-Bt corn and in which volumes. And we report back to the EPA on a regular basis the percentages of Bt and non-Bt corn by county across the U.S. Midwest. There's very little doubt that if you did not have an IRM strategy, that the European corn borer would become resistant. It's only a matter of when. And the use of an IRM strategy is to try to slow that down.
I certainly think the resistance management plans may help forestall resistance for at least some time. However, it's important to remember that the plans are basically built on some scientific experiments and a lot of mathematical modeling. They don't have a great foundation in practice.
Golden rice is also under scrutiny.
Golden rice is a technological solution for a really complex social issue, which is vitamin A deficiency. Vitamin A deficiency isn't a simple matter of just not having enough vitamin A. It's a matter of not having an adequate water supply, of not having enough food, of not having enough money to buy food, maybe of having bad living conditions, maybe of having a complicated cultural situation in which foods that contain beta-carotene aren't available or aren't supposed to be eaten by the people who most need them.
Many scientists feel that simply providing additional beta-carotene won't solve the problem, because the body has to be able to convert it to vitamin A.
The best and most efficient absorption of beta-carotene and the most effective use of beta-carotene comes in people who have plenty of fat and protein in their diets and are well nourished. It's difficult for people who have intestinal diseases, diarrheal diseases, to absorb beta-carotene because their intestinal tract isn't working well. So we have to ask the question, will beta-carotene in rice solve that problem? I don't know.
I think the key thing is this is not a silver bullet. Yeah, nobody is saying that this is the solution to the vitamin A deficiency problem. To solve vitamin A deficiency, the world, and particularly the countries where this is a serious problem, are going to have to implement a variety of strategies. And yes, the ultimate solution is for these people to have a balanced diet. But I don't see where that's any reason for not making rice a potential contributor to that balanced diet.
As the technology around GMOs advances, so does the controversy. Scientists are able to insert foreign genes into animals, too, using techniques like nuclear transfer, the science behind cloning. Dolly was cloned from a cell from an adult sheep, but like the sheep behind me, Dolly is not transgenic. Transgenic animals must carry a foreign gene. The ability to create transgenic animals has opened the door to countless applications. Today, the industry of farming, as in pharmaceutical farming, is booming. GTC Biotherapeutics is a state-of-the-art dairy farm in central Massachusetts. But milk isn't the only product. The business here is therapeutic human proteins.
Our lead program is something called antithrombin 3. Antithrombin is a protein normally in people's blood, and it participates in the coagulation process. It has both anticoagulant and anti-inflammatory properties. We have been able to make that in the milk of transgenic goats.
Goats are used because they're sociable and easy to work with, have short gestation periods, and are unrivaled in their ability to produce milk.
We're taking advantage of the mammary gland's abilities to make liquid proteins. Whether it's a goat or a human or any mammal species, milk is a rich source of protein for the offspring. And what we're doing is using DNA programming simply to get that mammary gland to make a protein of therapeutic interest to man.
To create a transgenic animal, a DNA construct containing a transgene can be introduced into early-stage embryos by one of two different methods. One is microinjection, where the transgene construct is inserted into an early-stage embryo with the nucleus intact. The other is nuclear transfer. In nuclear transfer, scientists first combine the human gene coding for antithrombin 3 with the promoter for the beta-casein gene.
A beta-casein promoter is what ensures that this protein, once it is produced in the animal, is produced only in its milk and not somewhere else in the animal's body. We're taking this genetic construct that I've told you about, we're putting it into cell lines and growing it up in cell lines. We're enucleating the egg from a donor mom and putting the cell line material into the egg.
The egg containing the transgene is transplanted into a surrogate mother and allowed to develop as in a normal pregnancy.
As that egg develops into a fetus and grows into an animal, we know that the animal that's being born will be transgenic. And therefore it offers a more deterministic way of knowing when we'll achieve a transgenic animal than the microinjection method offers. And thereby we're able to be more schedule predictive with the technology.
Goats reach sexual maturity at about 7 months. Lactating females produce the milk, which is collected like in any dairy. To recover the biologically active proteins, the milk then goes through a series of purification steps.
It's in studies right now, about to enter an efficacy study, to treat the condition known as hereditary deficiency, those people that don't make enough of antithrombin in their own blood and therefore are prone to blood clots.
Genetically altering animals to produce human proteins has sparked concern, not only about the safety of the proteins produced, but the general welfare of the animals.
After all, there's no people for the ethical treatment of soybeans, but People feel a little bit differently about animals and the potential impact of genetic engineering on their health and well-being and so on. The primary issues are really ethical ones. Are we comfortable with an animal that is, in essence, part human?
We are inserting a single gene in such that the desired protein is produced in the animal's milk. It's stable, it stays there, but it's still just a goat. It's not a hybrid, it's not a humanoid, it's not a mixed breed, it is still a goat.
Producing transgenic animals is one of the fastest growing biotechnology industries in the world, and countless varieties of genetically modified plants are under development as well. Biotechnology and the manipulation of genes is not new. What's new is that scientists can now precisely and quickly move a single gene between species.
Genetic engineering has all the potential to be good, and it has all the potential to be misused.
It's a technological miracle from the standpoint of what it can do, but it raises all kinds of social questions. And those social questions are important ones and deserve a great deal of attention, and they have to do with who controls the food supply, whether the foods are labeled so that people know and can make a choice about genetically modified foods, what the issues are in intellectual property rights.
Then there are broader issues about, is this use of genetic engineering actually something that's making agriculture more environmentally sustainable or less sustainable? Is the crop going to encourage more vast monocultures of corn or soybeans?
There's a huge potential of what you can use biotechnology for. And it's not just plants, it's animals, it's medicine. You can do a lot to the benefit of mankind with the proper focusing of some very good tools.
Funding for this program is provided by Annenberg CPB to advance excellent teaching. For information about this and other Annenberg CPB programs, call 1-800-LEARNER and visit us at www.learner.org.